Hello, my name is Mary Kathleen Shaw, and I am doing my educational presentation on inheritable genetic mutations. This is for Grand Canyon University for Bio 510, a comprehensive overview of cell and molecular biology. So in this presentation, I will be focusing on three classes of inheritable human mutations. The first mutation I'm going to be looking at is point mutations. Point mutations are changes in the nitrogenous base that's exchanging one base for another. Another class of inheritable human mutations are chromosomal mutations. That's a change in the location of DNA sequence. So the sequence is moved from one location to another. Another class is copy number variation. That's a change in the number of repeated sequences. So the sequences are repeated more frequently than normally would be found. So within these three classes, there are several types of mutations, and I'm going to focus in on one type of each of these classes of mutations. So why is it important to study genetic mutations, especially inheritable genetic mutations? Well, it's important because at the conclusion of the Human Genome Project, and also with all of the genetic research in the past 50, 60 years, scientists have discovered many mutations, and they have found pinpointed exactly where those mutations are located on the chromosomes. But so far, they have been unable to change those chromosomes and change those nitrogenous bases to be able to prevent these diseases from happening. So it's important to study it so that we can further the research. So genetic testing, whether that's prenatal genetic testing or for example, testing in adult females, seeing if they have the BRCA gene to know how they can prevent cancer and have preventative treatment. So it's important to study this in order to prevent and treat diseases. And also, of course, as teachers, we want to inspire the next generation. We want to inspire the next generation of scientists to be involved and study these topics. So who is the intended audience for this presentation? It's for AP Biology students. These students will have been well into the year and understand DNA structure and function, understand mitosis and meiosis, and will understand the topic that we are discussing. So inherited genetic mutations will be introduced and explained through this PowerPoint. The presentation will focus on three types of genetic mutations and the diseases that result. And we are explaining these genetic mutations. I'm going to relate it to a peanut butter and jelly sandwich recipe. What we are not talking about is mutations in other organisms. We are also not talking about genetic mutations that are not inherited, meaning that they are non-viable. So there are a lot of mutations that do not produce a living organism. So we won't be talking about those either. Okay, so DNA is like a recipe. It's like a recipe to make a person or make any kind of organism. So DNA is our recipe to make us. So here is a recipe for a peanut butter and jelly sandwich. So you can see that the utensils needed are a knife and a tablespoon. You will need 2 slices of sandwich bread, 2 tablespoons of grape jelly, 2 tablespoons of peanut butter, and then you're going to spread the peanut butter on one slice of bread, spread the jelly on the other slice of bread, and then put those 2 slices of bread together to form a sandwich. So the first class of genetic mutations that we're going to be looking at are point mutations, and the specific type is substitution. And again, there are several types of point mutations, but we're only looking at substitution. So in substitution, one base is incorrectly substituted during DNA replication. So the complementary base in the mRNA is different, and therefore, when the codon is read, a different amino acid is made. So in this original sequence, you can see here that there's a thymine, but then in the point mutation, instead of it being a thymine, now it's a cytosine, and that will that codon will code for a different amino acid. So if we're thinking about our peanut butter and jelly recipe, one thing that we can think of is we're going to substitute the word pepper instead of the word grape. So in the recipe, you can see that instead of 2 tablespoons of grape jelly, we will be using 2 tablespoons of pepper jelly. And although only one word is changed, the sandwich will certainly taste different. So one example of a single point mutation is sickle cell anemia. Sickle cell disease is a group of inherited red blood cell disorders caused by a single point mutation. Sickle cell anemia is the most frequent and the most severe form of the disease. So, in sickle cell anemia, GAG is changed to GTG and the sixth codon of the beta globin gene, and that replaces the glutamic acid for valine. So, the protein that's formed is long and inflexible and causes the red blood cell to change shape and look like a sickle. So, what are the symptoms of sickle cell anemia? Just that one, it's incredible to think that we have 3 billion base pairs, and this is one single substitution, and how it is such a devastating disease. So, what are the symptoms of sickle cell anemia? Anemia, that is, the red blood cells do not live as long as normal red blood cells. There are episodes of pain because the change in the shape of the red blood cells clogs up the capillaries, the arteries, and the veins, and causes severe pain. Also, swelling in the hands and the feet for the same reason. That's the sickle cells are blocking the flow of the blood. Also, frequent infections and vision problems. So, sickle cell is a homozygous recessive disease. So, to inherit this disease, each parent must be a sickle cell carrier. The carrier parents will not exhibit the disease, but the offspring will. And interesting and a bit of a scientific mystery is that carriers are better able to fight against malaria. It has to do with how the parasites try to enter the red blood cells, and they cannot enter the red blood cells the way they would like to in carriers of sickle cell disease. Okay, so the next type of mutation I'm going to talk about is chromosomal mutations. That's inversions. So you can see here is a picture of a chromosome, and a section of the DNA is flipped. It's inverted. So when I'm thinking about my peanut butter and jelly sandwich recipe, if you look at this recipe closely, you will see that 2 slices of sandwich bread has been flipped upside down and moved down into the recipe further. So this would be a good example of this type of inversion. So in the recipe, the ingredient 2 slices of bread has been flipped upside down, moved to a different location. So the portion of the recipe can no longer be read correctly. So it can no longer read 2 slices of sandwich bread. So what you end up with is peanut butter and jelly, but no bread. So it dramatically changes the recipe. Okay, an example of this type of mutation is FG syndrome. FG syndrome is an X-linked recessive disorder found in males. The mutations are found in the MED12 gene on the X chromosome. So some carrier mothers exhibit similar facial features, but they do not have intellectual disabilities. So FG syndrome is named after the initials of the first patient who was diagnosed with this disorder in 1974. It also has a name named after the 2 scientists who diagnosed it, Opitz and Kaverga. That's the Opitz-Kaverga syndrome. So males will be the ones who have this disorder, and it exhibits through intellectual disability, distinctive facial appearance, broad thumbs and great toes, a large head compared to the body, brain abnormalities in the corpus callosum, heart defects, and seizures. There's a he passed away in 2009, but there was a very famous man. He was called a mega savant, incredibly intelligent, but he had a lot of other disabilities, but his name was Kim Peck. He had FG syndrome, and he was the inspiration for the movie Rain Man, which won a lot of Academy Awards. Okay, so the third type of mutation that I'm going to talk about is copy number variation. This is an expanding trinucleotide repeat. So the normal number of repeated trinucleotide sequences is expanded. The codon is read correctly, but the amino acid that it codes for is repeated, changing the shape of the protein. So you can see here is a repeat, and it's only repeated so many times. And so this is the structure of the protein, but here it is repeated over and over again, and it completely changes the shape of the protein. So when I'm thinking about my peanut butter and jelly sandwich, instead of using 2 slices of bread, I'm going to try to make the sandwich with 100 slices of bread. So that will absolutely change the recipe. So try to imagine a sandwich with 100 slices of bread. So an example of this is a very devastating disease called Huntington's disease. It is caused by repeated CAGs. The length of the repeat affects the onset and the severity. So less than 35 repeats is normal and does not lead to Huntington's disease. The most common is about 40 to 50 repeats of the CAG, and it produces the classic Huntington's disease symptoms, and it begins in midlife. Greater than 50 repeats is juvenile Huntington's disease, and the symptoms begin as early as age 3. One thing about this disease, going back up to the most common are the 40 to 50 repeats that begin in midlife, meaning that those who are going to get it have already had the opportunity to reproduce and pass those genes along to the next generation. So it is autosomal dominant. So only one parent needs to have this defective gene. And some of the symptoms are uncontrolled jerky motor movement called chorea. So its original name was Huntington's chorea. Now it's called Huntington's disease. Gait disturbances in walking, dystonia, which is uncontrollable muscle contractions, neuropsychiatric symptoms like depression and anxiety. And in juveniles, if it's juvenile onset, it's just the severity is much greater. And there's also an incident of seizures. So as I am concluding this presentation, there are a lot of great resources out there, but one thing that I found especially helpful in researching these mutations was the societies that support these diseases and support the research. So the Huntington's Disease Society of America, as well as the Sickle Cell Disease Association of America. And this is a list of all the references that also includes the links to the material that I used. I hope you enjoyed this presentation.